Students, welcome to Sunil's tutorial. I'm Sunil Mirwani and today we'll be doing this chapter called as Genetic Basis of Inheritance. Let's understand what is backcross. Let's understand the meaning of the word backcross. Now what is backcross? Backcross is defined as the cross between F1 hybrid is defined as the cross between F1 hybrid and any of its parents. That is what is backcross, right? Now any of its parent, it could be the dominant parent or the recessive parent. That means F1 hybrid means, now first of all let's understand from where will I get F1 hybrid. F1 hybrid means it is formed from a cross between two parents. F1 hybrid means it is formed in a cross between, suppose if I take two characters as tall and dwarf, pure tall, that is TT, and pure dwarf. That is TT, right? Now, that is how you will get F1 hybrid. Now let's consider dominant backcross. Right? In dominant backcross, it is a cross between the F1 hybrid and the dominant parent. Right? The cross between F1 hybrid and the dominant parent, that is TT, is called dominant backcross. Fine, do we get this in here? So do we understand what is dominant backcross? Now, all the resulting offsprings will possess dominant characters in this case, right? All the resulting offsprings will possess dominant characters. That is, you will get 100% dominant characters. But this does not give you an idea about the genotype of the individual, right? I will see a dominant character, that is the phenotype is going to be dominant character, but we don't know what the genotype is. So let's see that. But this does not give any idea. This does not give any idea about the genotype of the individual. Fine. Whether the genotype is TT, both the capital T or one capital T and small t, so the backcross cannot be called as a test cross. So backcross cannot be called as test cross. Right? Let's try to understand this with the help of a diagram. Phenotype, you have F1 plant. This is tall, and you have tall dominant pure parent. Tall dominant pure parent. Right? The genotype is This is hybrid tall, therefore this will be TT. This is pure tall, therefore this will be TT. Gametes. This will give you two different types of gamete, T or small t. This will give you only one type of gamete, that is capital T. Right? Now let's see the checkerboard. Now, in one case you are going to have both the gametes, that is T and T, right? And the other is going to be capital T and small t. You will have male, female, right? Now in this case you will get TT, TT, capital T, small t, capital T, small t. So this is tall. All four of them are going to be tall. Two of them are going to be pure tall and two of them are going to be hybrid tall. Right? So this is going to be a back cross between F1 generation and dominant parent. Now let's try to see a back cross with the recessive parent. Right? Recessive back cross. What is recessive back cross? It is a cross between the F1 hybrid and the recessive parent. It is the back cross between F1 and recessive parents. This is what is recessive back cross, right? So in recessive parents, that is small tt, right? All the resulting offsprings will show dominant character, and the all the resulting offspring show dominant character to the extent of 50% and recessive character to the extent of 50%. So they will show dominant as well as recessive character, right? Therefore, the ratio of tall is to dwarf, is 1:1, right? Which gives the clue that the individual is heterozygous. The individual is heterozygous, right? Therefore, such a back cross can be called as a test cross. Such a back cross can be called as test cross, right? So that's how you do a recessive back cross. Let's try to understand this with the help of a diagram. Parents are pea plant, right? Then let's see the phenotype. The phenotype is F1 plant, tall, which I'm going to cross with dwarf recessive. Parent, right? The genotype tall, TT, dwarf, TT, right? This will give me gametes. This will give you gametes T and T. This will give you gamete only T, right? Now let's make the checkerboard. So in one case you are going to have only TT, in the other case you are going to have TT. Male, female, right? This will give you TT, this will give you TT, T and T and TT. So this will give you tall, dwarf, tall, dwarf. Right? 50% of the offsprings are going to be dwarf. Fine, do we get this thing here? Why do I need to study about back cross? Right? Significance of back cross. What is the significance of back cross? Now it is used in horticulture or animal breeding. It is used in horticulture or breeding. That's one of the reasons why we study about back cross. It is an easier, quicker method of desirable trait in a pure homozygous condition. It is easier and quicker method to get your desirable trait in pure homozygous condition. So if I want a particular trait, then it's easier to get that specific. I want in the F1 generation or the F2 generation, I want a specific trait, then it's very easy to get that using a back cross, right? So those are the two reasons why we study about back cross. Next, let's study about test cross. Now what do I understand by test cross? Test cross is defined as the crossing between F1 hybrid with its homozygous recessive parents, and the progeny of the test cross is called as the test cross progeny. So you would have F1 and the recessive parents. Fine, do you get this in here? It's your similar to recessive back cross. Right, do you get this in here? So when you consider recessive back cross, that's called as your test cross. We already wrote that, right? I'll just write this once again. What is test cross? It is defined as the crossing of F1 hybrid. It is defined as the crossing of F1 hybrid with its homozygous recessive parents. With its homozygous recessive parents. Right, that is what is test cross. And the progeny of the test cross is called as the progeny of the test cross is called as test cross progeny. Test cross progeny. Right? That is what is test cross. Now, recessive back cross is called as test cross. So I could say that recessive back cross is called as test cross. So whether I say recessive back cross or test cross, it's the same thing. Thus, test cross is the back cross, but reverse. Thus, a test cross is a back cross, but reverse is not always true. Thus, test cross is, you will get this as true or false, is a back cross, but the reverse is not always true. Right? I'll just show you the diagram again. You would have parents, that is your pea plant. Phenotype is going to be F1 hybrid tall, heterozygous F1 hybrid tall, or cross with recessive parents, homozygous dwarf. Recessive parents which are homozygous dwarf. Right? That's my phenotype. Genotype will be nothing but TT and TT. Gametes is going to be TT, and here you are going to have only T. Right? Now, let's see the checkerboard. This is going to be, say, TT. This is TT. Consider one to be male, the other to be female. Right? So TT, TT, TT and TT. That's going to be my checkerboard pattern. Right? So in this case, what we see that the progeny of the test cross shows both dominant as well as recessive characters in the ratio of 1:1. Right? Then the individual can test, the individual under the test is heterozygous if the progeny of the test cross shows dominant character and the individual is homozygous. So you will come to know whether the individual is homozygous or heterozygous on the basis of this. Right? If you get 1:1 ratio, in that case then the individual is going to be heterozygous. Right? So I can say that the progeny of the test cross the progeny of the test cross shows both dominant and recessive characters. Right? The progeny of the test cross shows both dominant and recessive characters in 1:1 ratio. Right? If I get 1:1 ratio, then the individual under test is heterozygous. Then the individual under test is heterozygous. If I get both dominant and recessive characters in the ratio of 1:1, then it is heterozygous. If the progeny of the test cross shows only dominant characters, if the progeny of the test cross shows dominant characters, if the progeny of the test cross shows dominant character, then the individual tested is homozygous. Then the individual tested is homozygous. That's how you find out whether the individual is homozygous or heterozygous, and that's specifically the reason why this is called as a test cross. Now we could do a test cross with dihybrid. We could do a dihybrid test cross. Let's consider a dihybrid test cross. Let's try to understand what is a dihybrid test cross. It's a cross between the F1 dihybrid and its double recessive parents. What is dihybrid test cross? The cross between F1 dihybrid and its double recessive parents. Double recessive parents is dihybrid test cross. Fine, do we get this in here? Now why is it done? It is done to test the correctness of Mendel's law of independent assortment. Right? It is done to test the correctness of Mendel's law of independent assortment. Where he said that the gametes remain in their true form and they show themselves in F2 generation, right? Because if Mendel's principle of independent assortment is correct, then the F1 hybrid should show 4 types of gametes in equal proportion, and the recessive parent will produce only 1 type of gamete. Right? In dihybrid, I can say if Mendel's principle of independent assortment is correct, then F1 hybrid should produce 4 types of gametes. In equal proportion. F1 hybrid should produce 4 types of gametes in equal proportion, and the recessive parent shall produce only 1 type of gamete, and the recessive parent shall produce only 1 type of gamete. Right? Thus, the progeny of this cross will show 4 types of phenotype in equal proportion. Right? So, if you have 4 different types of gametes and only 1 type of gamete of the recessive parent, then I can say therefore the progeny of this cross will show, the progeny of this cross should show 4 types of phenotypes in equal proportion. 4 types of phenotypes in equal proportion. That's what I should get in a dihybrid test cross. Let's try to check whether you can get this. Let's see a dihybrid test cross. Dihybrid test cross. Right? Parents are going to be pea plants. Right? Then your phenotype. Now, Now, here you are going to consider round and yellow. Which will cross with wrinkled green. Genotype. This is your F1 hybrid. So round will be RR and yellow will be YY. Capital Y, small y. Wrinkled green is going to be RR, YY. So here you can see that you have a cross between hybrid and a pure gamete. Right, now let's see the gametes that will be produced. Now this is going to produce 4 different types of gametes. That is going to be RY, RY, RY and RY. These are the 4 different types of gametes that will be produced by the F1 hybrid, and the wrinkled green is going to produce only one type of gamete. Right? Now let's check the test cross offspring. Test cross offspring by checkerboard. Right? On one of the axes you are going to have 4 different types of gametes. That is, you are going to have RY, capital R, small y, small r, capital Y, small r, small y. On the other axis you are going to have RY, only one type of gamete. Right? One of the plant is considered as male, the other is considered as female. Right? So in this case, what you will get is you will get RRYY. That is, you will get hybrid round yellow. Here you will get RRYY. So here you are going to get hybrid round green. Then here you will get RRYY. You are going to get hybrid green. You are going to get hybrid wrinkled yellow, and finally you will get RRYY. That is hybrid wrinkled green. Now, since there are 4 different types of output, here you are getting round yellow. This is round green. This is wrinkled yellow, and this is wrinkled green. Since there are 4 different types of output, what is the possibility of each type of output? The possibility of each type of output therefore is 25%. 4 different types of outputs are possible. That means the possibility of each type of output is 25%. So in that case, my dihybrid test cross ratio should be dihybrid test cross ratio should be 1:1:1:1. That will be my dihybrid test cross ratio. Next, why do I need to study this? Significance of test cross. Why do we need to study this? Right? Now, the reason why I would study this is because it gives genotype of F1 hybrid and also proves one of the parent of F1 is homozygous recessive. So you come to know whether you are doing a cross with a recessive parent or a dominant parent. First, it gives genotype of the F1 hybrid. Tells you what was the genotype in the F1 hybrid, and also proves that one of the parent It also proves that one of the parent of F1 is homozygous recessive. That's the reason why you have the properties coming back, all the 4 different types of properties, right? Why would I use this in practical life? Because it's a rapid method of improving the variety of crop plants, right? It improves, it is a rapid method of improving the varieties of crop plants. So if I have to improve the variety of crop plants, I would use this. It proves the law of heredity, both the laws, that is the law of segregation as well as the law of independent assortment. The gametes remain, they do not mix, and then they show themselves back in the F2 generation. So it proves the laws of heredity. Which are the laws of heredity? That it proves both laws of law of segregation and law of independent assortment. Both the laws are proved in this, right? And last, more homozygous individuals with desirable characters are obtained using test cross. More homozygous individuals with desirable characters are obtained. So that's your significance of test cross, right? Okay, we'll stop this here for the day. Thank you very much.